Hello, I'm Kevin Davis, editor of BioIT World, here in Providence, Rhode Island at the Convention Center. CHI's Exploring Next Generation Sequencing meeting is going on behind me. And joining me is an old friend, a former colleague, Laurie Goodman. We were at Nature Genetics together. Laurie is the founding editor of Genome Research, one of probably the leading journal in the whole genomics field right now. And her latest gig, which is why she's here with me, is as the Public Information Officer for the Beijing Genomics Institute of Shenzhen. Laurie, welcome.
Thank you very much. Nice to see you again.
Indeed. Laurie spoke today with the head of the BGI Shenzhen, unable to make it because of visa problems. Second time we've tried to bring him over here. But tell us a little bit, Laurie, about the Genome Center in Shenzhen.
Specifically in Shenzhen, that has now become, of the BGI, there are 3 BGIs, that has now become the main sequencing center for the Beijing Genomics Institutes. And it is especially focused on next generation sequencing technology, and they're very keen on pushing the envelope with the latest and most effective sequencing that comes out. So they're always watching for that sort of thing.
You said in your talk that the Shenzhen operation is the 3rd largest sequencing facility in the world, the largest in Asia, probably by some margin. What's the current setup in terms of next-gen platforms there?
So they now have 18 Illumina genome analyzers. They have 2 454 Roche FLX sequencers, and they have 3. And they have 2 of the solid, AB solid sequencers.
And we'll come to their sort of flagship project in just a second, but what's sort of the range of projects that are going on there?
So they actually have a huge range of projects. They have work that is in bacterial and viral genetics. That's a special focus there because of a lot of the infectious agents in China.
SARS and...
Yes, exactly. And agricultural genomics is very big. They, up at the BGI in Beijing, they sequence the rice genome. And but they're continuing with working on rice, and also have been doing cucumber and a bunch of other collaborations internationally with a variety of crops.
Panda?
Panda was announced this year, and they're also working on a variety of animals. Basically, they are very can-do. If it's of use, if it's cute, if it causes diseases, they're very interested and able to do a lot of that sequencing and get it done.
Now, about 12 months ago, maybe just under, the folks there announced the sequencing of the first human genome from Asia. And as you presented this morning, that work is provisionally accepted at Nature magazine, which is great for them. Tell us a little bit, obviously it's not a huge amount of detail because the work has yet to be published, but just an overview of what they've accomplished.
Well, the interesting thing about this is it is the first genome to be done with this short read genome sequencing. It was all done on Illumina genome analyzers. They got about a 34-fold depth in sequencing, which is very high, but it's important to get that type of depth because of the short reads. They discovered, not sure how many, 400,000 SNPs, and they have tested the accuracy of that in comparing it to genotyping, and had 99% coverage. Also, the SNPs that were not consistent, they were able to sequence that using PCR, and the resequencing was actually more accurate. 80% of the SNPs were confirmed from the sequencing than from the gene chip, so the errors seem to be in the genotyping. You
said 400,000. Did you not say 3 million in the talk?
You know, I may have. I'm blanking on the numbers a lot, and I guess 13% of those were not found in dbSNP. So they're finding some new things, but until you really have more genomes, it's difficult to say how rare those are, how unique those are.
And what can you say about the donor of the DNA?
So the donor is from the 30% population in China, the Han Chinese. that is the greatest population of the different Chinese ethnic groups. China has a huge number of ethnic groups. Some of them are very minor, and the Han Chinese is one of the groups that comes from a very prestigious ancestor and is, in addition to being well respected. So.
And plans for other Asian genomes, perhaps as part of the 1,000 Genomes project?
Yes, so, and Shenzhen will be doing all the sequencing work. They're responsible for all of the Asian genomes that will be sequenced in the 1,000 Genome Project, and that will be about 100 individuals. And they'll, again, be using this next generation sequencing.
So these figures will be a little out of date because the costs have changed and the throughput has changed, but for the work that was done for this, which I guess was the sequencing was done last year because that's when it was announced, you said 5 machines and the cost?
Was about $500,000 US, and basically they can get the reads that they need in, well, they said 1 to 2 months, so maximum 2 months.
These days it'll be a lot quicker, I'm sure. Another fact about BGI Shenzhen is that it was selected by Genome, or Genome, or we still don't know how to pronounce the... Genome, that was the one. The company set up by, co-founded by George Church to provide sort of bespoke sequencing, whole genome sequencing for pretty well-heeled individuals. $350,000 was the original price tag. Can you say anything about how BGI came to be selected? What does that say about the facilities and the quality and the price of what they're doing?
Well, one of the things about BGI is, first of all, the people are very hardworking and very geared toward innovation. So if something's not working, they work very closely with the company. They help design new processes in order to improve the sequencing accuracy. They, for this project that they just did, they designed the SNP method that had to be specialized for this short read sequencing. And in addition, because they're so dedicated to the sequencing, they're the third largest sequencing, as you mentioned, in the world, they are well prepared to carry out these types of sequencing. And because they're very proactive and very interactive with collaborations, they're an excellent choice for.
Well, and you had your work cut out, I guess you were saying, editing this massive manuscript down to meet nature's rather strict page constraints?
Yes, well, when things are being presented to nature, you want all the information in there because it goes out for review, it comes back, they want more information, which, of course, reviewers are supposed to want more information. So by the time you get to the point where the paper is now ready to be accepted, this paper was about 8,000 words, and so it needed series cutting. Nature's limit for articles is 3,000 words. So, but the nice thing is online supplementary material. So, a lot of the data that had to be removed for space is. Well,
as a former journal editor, that was obviously easy work for you, I'm sure.
Yeah, and fun. It's a joy really working with the folks in Shenzhen.
Well, we hope next time we hold this meeting, we'll find Wang Jun's visa problems will finally get sorted out and we can have him over. Otherwise, we'll see you in San Diego. So, Laurie Goodman, thanks so much.
Thank you very much.
Kevin Davis for BGI World. See you again.